In the previous class, we were discussing about cloning vectors, and we also learned one example of the cloning vector, which is plasmid. We have learned about the artificial plasmids PBR 322, PUC 19, PUC 101. And in today's class, we will be learning other examples of the cloning vectors. The second example of cloning vector is bacteriophages. Now, what are bacteriophages? These are the virus which attack bacteria. So, bacteriophage is a virus which attacks bacteria. Now, what we can observe is the bacteriophage, when it attacks the bacteria, the virus DNA or the bacteriophage DNA enters into the bacterial cell, and in the bacterial cell, the viral DNA or the bacteriophage DNA is undergoing replication. Right. Now, what we do is, we modify the bacteriophage DNA. We are modifying the bacteriophage DNA such that the gene of interest, here I have highlighted with a black color. So, here we are inserting the gene of interest. Now, this kind of the modified viral DNA which is having the gene of interest. Now, this phage with having the modified viral DNA consisting of the gene of interest or the foreign gene or the desired gene. So, when it is infecting the bacterial cell, so this kind of a phage is allowed to infect the bacterial cell. So, what happens is, along with the viral DNA, the gene of interest is also replicating. So, here we can observe even the gene of interest is also replicating. So, that is how we can able to introduce the gene of interest into the recipient cell or the host cell by using the bacteriophages. So, by attaching the gene of interest to the viral DNA and by allowing these kind of bacteriophages to infect the bacterial cell or the recipient cell or the host cell here, we can able to clone the gene of interest. So, along with the viral DNA, even the gene of interest is also cloned, increased in its number, and even the gene expression also takes place in the bacterial cell. The gene of interest is undergoing gene expression, transcription, translation in the bacterial cell. So, this is by using the bacteriophages, we can able to transfer the desired DNA fragment into the host cells. Right, the next type of vector which we are using is cloning vector is Cosmids. Now, Cosmids, these are the hybrid vectors. Now, these hybrid vectors, they are made up of genes from two sources. Now, let's understand what are those two sources. So, this is a black color which I have represented. This is a plasmid. Now, to this plasmid, what we do is, we are adding cohesive sites or the sticky ends or the cos sites of the lambda phage. So, this is the lambda phage cos site. So, simple. Here you can notice from the lambda phage is a bacteriophage again. So, from the lambda phage, what we are taking is the cohesive sites or the sticky ends or the cos sites, and we are binding these cohesive sites or sticky ends or the cohesive ends, cos sites, with the plasmid. So, this plasmid is obtained from a bacterial cell. So, we are attaching the plasmid with the cohesive site, and cos site plus plasmid becomes a cosmid. So, this cosmid is also used as one of the important cloning vector. So, to this cosmid, what we do is, because it has a sticky end, So we are adding the gene of interest here such that it will be easily binding with the cohesive site and a recombinant DNA is formed. So simple. The third cloning vector is Cosmid. Cosmids are hybrid vectors. They are made up of genes from two sources. So what are those two sources? From the lambda phage, we have extracted the cohesive sites or the cos sites or the sticky end, and we are attaching with what? Plasmid, which is extracted from a bacteria. So plasmid plus cos site is Cosmid. Now Cosmid is acting as a cloning vector. So to this Cosmid we will be adding attaching the gene of interest. So this is about the next variety, which is Cosmid. Let's go with the other type of the cloning vector, which is artificial chromosomes. Now what are these artificial chromosomes? Artificial chromosomes are the DNA. We are preparing these cloning vectors artificially in the laboratory, where we attach the DNA with centromeres, we add ORI regions, we add telomeres, and we add selectable markers. That means what? To a DNA we add ORI region, we are adding the ORI region, origin of replication region, we are adding centromeres, we are adding telomere region. Why telomeres? So that the DNA should be become in the linear fashion, it should be in a linear fashion. So that is the reason why we are adding telomeres, and we also add selectable marker. Okay, DNA ki manam centromeres, ORI, telomeres, selectable marker add చేసి artificial chromosomes ni తయారు చేస్తాం. So we are constructing these artificial chromosomes by doing this sort of a procedure. Now let's take some examples of the artificial chromosomes. The first example is yeast artificial chromosomes, and this is very important, and it is a widely used cloning vector, especially for cloning large DNA fragments. Yeast artificial chromosome can carry large pieces of gene of interest, large desired DNA fragments, and not only that, even they can able to clone, increase the number of gene of interest in the recipient cell. So that is the importance of yeast artificial chromosomes. And these chromosomes were widely used in the Human Genome Project. When mapping the human genome, so these yeast artificial chromosomes were widely used. The second type of the artificial chromosome here is bacterial artificial chromosomes and human artificial chromosomes, mammalian artificial chromosomes. So these are all the artificial chromosomes. So artificial chromosomes are prepared by adding DNA with the centromeres, with the ORI region, telomeres, and selectable markers. Examples: yeast artificial chromosome, bacterial artificial chromosome, human artificial chromosome, mammalian artificial chromosome. In that, the yeast artificial chromosomes are widely used, especially when the desired genes are very large. Not only they carry the large desired DNA fragments, but they are also useful for cloning, for increasing the number of the desired DNA fragments into large numbers. Right. So this is the importance of EAC. Next one is, the next type of cloning vector is phagemids. Right. Now here phagemid is again a hybrid vector. It is also made up of genes from two sources. The first source is bacteriophage DNA, and along with that plasmid DNA. So a bacteriophage DNA is attached with the plasmid DNA, and what is formed is phagemid. Right. The next type of cloning vector which we use is transposons or mobile genes or jumping genes. So these transposons or mobile genes or jumping genes are the DNA sequences which usually jump from one location to the other location. Now these Transposons can also be used as a cloning vectors. And another type is shuttle vectors. Now what are these shuttle vectors is, these kind of vectors have two types of origin of replication and two types of selectable markers, such that these vectors can be used for both eukaryotic cell and prokaryotic cell. So these shuttle vectors are used for both eukaryotic and as well as prokaryotic cell. So these are some of the examples of the cloning vectors. So NCERT has highlighted examples such as plasmids and artificial plasmids, PBR 322, PUC 19, PUC 101. And the second important thing NCERT has highlighted is bacteriophages. And the third thing is about the cosmids. And the fourth thing is artificial chromosomes. Only examples we are supposed to learn, not about the detailed procedure how they are constructed. So only the examples are needed. So these are the examples for the cloning vectors.